Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 51. The topic is Fermentation and Anaerobic Respiration. Discussing agenda for the topic is fermentation and anaerobic respiration enable cells to produce ATP without the use of oxygen. Types of fermentation, in which we will discuss alcoholic fermentation and lactic acid fermentation, and then finally we will compare fermentation with anaerobic and aerobic respiration. Lecture outcome. After watching and listening this lecture, students will know about various concepts related to life without oxygen. Introduction. Fermentation and anaerobic respiration enable cells to produce ATP without the use of oxygen. Now, we have already discussed the aerobic cellular respiration in detail, the electron transport chain, where oxygen is used as the final acceptor of electrons. Now suppose oxygen is not available, so is there another molecule which can act as the final acceptor of electrons? And what will happen when there is neither oxygen nor any other molecule which can act as the final acceptor of electrons? So what will happen? The process of glycolysis can take place without the need for oxygen. It can take place in the absence of oxygen as well as in the presence of oxygen. But the electron acceptors, the NAD, accepts electrons, it is converted into NADH, and then there must be some molecule to which this NADH delivers its electrons, because it will return back to glycolysis and once more accept electrons. So if there is no acceptor of electrons from NADH, then all NADH molecules will be occupied with electrons, and it will ultimately stop the process of glycolysis also, because the process of glycolysis require electron acceptors in the form of NAD. So when all NAD molecules are converted into NADH form and they are occupied with electrons, then the process of glycolysis will stop, because there will be no acceptor of electrons. So the organism or the cells that are respiring in anaerobic conditions must find some way to bring NADH back into NAD form. So in this lecture we will discover what are the different mechanisms which enables the cells to carry out the process of glycolysis in the absence of oxygen. Because most of the ATP generated by cellular respiration is due to the work of oxidative phosphorylation, which takes place in electron transport chain, where there are a series of reduction and oxidation reactions. So the bulk of ATPs are produced in electron transport chain through oxidative phosphorylation. Our estimate of ATP yield from aerobic respiration depends on an adequate supply of oxygen to the cell. So the availability of oxygen is very important for the continuation of electron transport chain, because oxygen acts as the final acceptor of electrons. When there is no final acceptor, that is when there is no oxygen, then all the carrier molecules will be occupied with electrons, and therefore this process of transfer of electrons will stop, and ultimately electron transport chain will stop working. So there must be some final acceptor of electrons. In organisms which respire aerobically, of course there is oxygen. So what if the organisms does not have the supply of oxygen, or if oxygen is poisonous for them? Say if the organisms are anaerobic, then what will happen? Which molecule will act as the final acceptor of electrons to continue the electron transport chain? Without the electronegative oxygen atoms without the electronegative oxygen atoms in O2 to pull electrons down the transport chain, oxidative phosphorylation eventually ceases. So when there is no oxygen present as the final acceptor of electrons, then all the carriers in the electron transport chain will be occupied with electrons, and there will be no one to accept electrons from them. So the eventual result will be the cessation of electron transport chain. However, there are two general mechanisms by which certain cells can oxidize organic fuel and generate ATP without the use of oxygen. So now we are talking about anaerobic respiration, which takes place in the absence of oxygen. So what if oxygen is not available? What will happen? Which molecule is going to act as the final acceptor of electrons in electron transport chain? The distinction between these byproduct two is that an electron transport chain is used in anaerobic respiration but not in fermentation. So the organisms which respire anaerobically, they can still drive ATPs using electron transport chain, but in that case, because the oxygen is unavailable, so there must be some other molecule which can act as the final acceptor of electrons. Now the electron transport chain is also called the respiratory chain because of its role in both types of cellular respiration. So the electron transport chain can operate both in aerobic and anaerobic conditions. But in anaerobic conditions, there must be some other final acceptor of electrons apart from oxygen. We have already mentioned anaerobic respiration, which takes place in certain prokaryotic organisms that live in environments without oxygen. So now we will discover how the process of electron transport chain occurs in those organisms who respire in the absence of oxygen. These organisms have an electron transport chain but do not use oxygen because they are anaerobes, so oxygen is not there as the final acceptor of electrons. So what else? Which molecule will act as the final acceptor of electrons in that chain? And oxygen performs this function very well. Now here is another point that even in anaerobic organisms the process of electron transport chain takes place because they have another types of atoms which act as final acceptor of electrons. But in that case the energy produced is extremely lower because this high electronegativity of oxygen enables this process to occur efficiently and generate maximum amount of ATPs. If other molecules act as the final acceptor of electrons, then still the electron transport chain will continue functioning, but it will not function as efficiently as if oxygen is present there. Some sulfate reducing marine bacteria, marine means living in salty environments, for instance use sulfate ion. So in these sulfate reducing marine bacteria, sulfate ion act as the final acceptor of electrons instead of oxygen. Operation of the chain builds up a proton motive force used to produce ATP, but hydrogen sulfide is made as rather than water in the aerobic respiration. In that case, the electron transport chain, finally the oxygen atom accepts electrons, then combines with two hydrogen atoms and form water. But in anaerobes, where the sulfate ion act as final acceptor of electrons, so it ultimately forms hydrogen sulfide. The rotten egg odor you may have smelled while walking through a salt marsh or a mud flat signals the presence of sulfate reducing bacteria. So the hydrogen sulfide actually smells like a rotten egg, and because in those organisms the final acceptor of electrons in electron transport chain is sulfate instead of oxygen, so the final product of electron transport chain is hydrogen sulfide, and therefore the rotten egg odor is on the observation in those marshy areas. Now, fermentation is a way of harvesting chemical energy without using either oxygen or any electron transport chain. Now, this is another type of respiration which takes place in the absence of oxygen, but in this case, not only oxygen is absent, but other electron acceptors are also present. So one thing is clear, that the organisms which respires in this way, in those organisms electron transport chain does not take place because there is no acceptor of electrons. Now, how can food be oxidized without cellular respiration? Remember, oxidation simply refers to the loss of electrons to an electron acceptor, so it does not need to involve oxidation. So how we define cellular respiration? Cellular respiration is a process in which food is oxidized and energy is released. So here the term oxidation is not used in a way that it is an oxygen dependent process, but because the loss of electrons is involved in this process, so therefore it is referred to as oxidation, and therefore it can take place even without the presence of oxygen. Glycolysis oxidizes glucose to two molecules of pyruvate. These are the end products of glycolysis. Pyruvate is a 3-carbon molecule and 2 pyruvate molecules are produced. So it means all the 6 carbon molecules in glucose are still intact in those 2 pyruvate molecules. So it means they can still provide energy. The oxidizing agent of glycolysis is NAD, and neither oxygen nor any electron transfer chain is involved. Now, because in those organisms the process of glycolysis is going on, which doesn't require oxygen. Now the process of glycolysis has occurred, and NAD has accepted all electrons and converted into NADH form. Now what will happen? This NADH form must give its electrons to another acceptor so that they can go back and participate in glycolysis again. If all the NAD molecules are occupied with electrons, then the process of glycolysis will also cease, will also stop, because there will be no acceptor of electrons. Overall, glycolysis is exergonic. It's a process which releases energy, and some of the energy made available is used to produce 2 net ATPs by substrate level phosphorylation. Once again, I am repeating, substrate level phosphorylation is a process in which one of the substrates, one of the intermediates of a biochemical pathway, provides electrons to NAD, provides electrons to ADP and converts it into ATP. If oxygen is present, then additional ATP is made by oxidative phosphorylation. Of course, if oxygen is present, then electron transport chain will take place, and oxidative phosphorylation will result in the production of ATPs, and the NADH molecules will also be freed from the electrons, because they will deliver their electrons to the electron transport chain. But glycolysis generates 2 ATP whether oxygen is present or not. That is, whether conditions are aerobic or anaerobic. It is not concerned with the presence or absence of oxygen. As an alternative to respiratory oxidation of organic nutrients, fermentation is an extension of glycolysis that allows continuous generation of ATP by the substrate level phosphorylation of glycolysis. Now, if the oxygen is not there as the final acceptor of electrons, it means the electron transport chain will not take place, and the NADH molecules will be occupied with the electrons. So there must be some other alternative where those NADH molecules can get rid of their electrons and return back and participate in the glycolysis. So this fermentation process is important for the survival of the glycolytic pathway. For this to occur, there must be a sufficient supply of NAD to accept electrons during the oxidation step of glycolysis. So NAD in this form is vital for the survival of glycolytic pathway, because it accepts electrons. If it is not available, if there is no acceptor of electrons, then this process will cease, it will stop. Without some mechanism to recycle NAD from NADH, glycolysis would soon deplete the cell's pool of NAD by reducing it all to NADH, and would shut itself down for lack of an oxidizing agent. So if all the NAD molecules are occupied with electrons, then ultimately the glycolytic pathway will stop working, because there will be no acceptor of electrons. So the need of the hour is to find some other way where NADH can get rid of its electrons and return back to this process, return back to this form, and once more participate in glycolysis. Under aerobic conditions, NAD is recycled from NADH by the transfer of electrons to electron transport chain. Remember that those NAD molecules which are produced in glycolysis, then in link reaction, then in Krebs cycle, all those NADH molecules deliver their electrons to the electron transport chain. They are freed, and then they can return and participate again either in glycolysis or Krebs cycle as the acceptor of electrons, but it only occurs in aerobic conditions. When oxygen is available, therefore electron transport chain takes place, and they have a way to free themselves with the occupied electrons. But what if oxygen is not available? What if electron transport chain is not working? Is there any other way where they can get rid of their electrons? We will discover soon. An anaerobic alternative is to transfer electrons from NADH to pyruvate, the end product of glycolysis. So one of the alternative for NADH to get rid of its electrons is to give it to pyruvate. Pyruvate is of course the end product of glycolysis. Now there are two types of fermentations. Fermentation consists of glycolysis plus reactions that regenerate NAD by transferring electrons from NADH to pyruvate or derivatives of pyruvate. The NAD can then be reused to oxidize sugar by glycolysis, means it can be reused to accept electrons once more, which nets two molecules of ATP by substrate level phosphorylation. There are many types of fermentations differing in the end product formed from pyruvate. So the pyruvate can change into different forms, but here we will discuss only two types of fermentations, which is alcoholic and lactic acid fermentation. Now, in alcoholic fermentation, pyruvate is converted to ethanol in two steps. The first step releases carbon dioxide from pyruvate. So remember, pyruvate is a 3-carbon molecule. When one of the carbon is liberated as carbon dioxide, then the molecule is left with only 2 carbons and it becomes acetaldehyde. In the second step, acetaldehyde is reduced by NADH2 ethanol. So then acetaldehyde accepts the 2 electrons from NADH. NADH is converted to NAD and it can go back and participate in glycolysis, and the end product is ethanol. This regenerates the supply of NAD needed for the continuation of glycolysis. Many bacteria carry out alcohol fermentation under anaerobic conditions. Yeast, which is a fungus, in addition to aerobic respiration, also carries out alcohol fermentation. Of course, alcohol fermentation will take place when there is no oxygen supply. Now, for thousands of years, humans have used yeast in brewing, wine making, and baking. The carbon dioxide bubbles generated by baker's yeast during alcohol fermentation allow the bread to rise. So you consume bread on regular basis in your daily life, and the bread is a kind of swollen. It is because of these carbon dioxide bubbles which are generated here in this process. Now here is just to shed some light on that structure. Here glucose enters glycolysis and 2 net ATPs are produced, and 2 molecules of pyruvate are produced. Remember, this 2 signifies 2 molecules of pyruvate. Now these 2 molecules of pyruvate will release 2 carbon dioxide molecules. The remaining 2 carbon portion is called acetaldehyde, and then the NADH, it gives its electrons to acetaldehyde and converts it into ethanol, and this NAD is available to participate in glycolysis again. Now the next type is lactic acid fermentation. During lactic acid fermentation, pyruvate is reduced directly by NADH. In alcoholic fermentation, first the pyruvate converted into acetaldehyde, and then acetaldehyde was reduced by NADH. But in case of lactic acid fermentation, pyruvate is directly reduced by NADH to form lactate as an end product, regenerating NAD with no release of oxygen. So mind the difference between the two processes. Now lactate is the ionized form of lactic acid. Lactic acid fermentation by certain fungi and bacteria is used in dairy industry, in dairy products, where milk, butter, yogurt, etc. is prepared to make cheese and yogurt. A complex series of fermentation and aerobic respiration pathways carried out by yeast and bacteria on cocoa beans is responsible for the production of chocolate. So means this process is also involved in the production of chocolate. What about lactate production in humans? So is lactate produced in humans? Previously we thought that human muscles only produced lactate when oxygen was in short supply, such as during intense exercise. So when there is intense exercise going on, then of course ATPs are used, and the process of glycolysis occurs over again and again, and because this process is occurring again and again and there is short supply of oxygen, the pyruvate cannot move ahead and participate into Krebs cycle and electron transport chain. So the alternative to get rid of NADH is to reduce pyruvate and form lactate. Research done over the last few decades, though, indicates that the lactate story in mammals at least is more complicated. So it is not that straightforward. Just said, there is some complications to the story, some mysteries to the story. There are two types of skeletal muscle fibers. One is red and the other is white. The red muscles oxidize glucose completely to carbon dioxide. The other white muscles produce significant amount of lactate from pyruvate made during glycolysis. So in white muscles, the pyruvate is converted into lactate, and it can only happen if NADH delivers its electrons to pyruvate, then it is converted into lactate. Even under aerobic conditions, offering fast but energetically efficient ATP production, energetically inefficient ATP production. So this process can occur even in aerobic conditions, where ATP production is fast because the pyruvate doesn't need to enter into a long chain of reactions entering Krebs cycle and then into electron transport chain, because the body is in urgent supply of energy. So the most efficient process in this condition is glycolysis, but of course in terms of ATP derived, it is not an efficient process because only 2 ATPs are produced. Net production is 2, but it can meet the urgent demand of energy. The lactate product is then mostly oxidized by red muscle cells in the vicinity. So when the lactate is produced by white muscles, the red muscles which lies around in the vicinity of the white muscles, they oxidize this lactate, with the remainder exported to liver or kidney cells for glucose formation. Now remember that the lactate can be converted back into glucose in the liver. Through which process? Through Cori cycle, C-O-R-I. Through this cycle, the lactate produced can be converted again into glucose or glycogen. Because this lactate production is not anaerobic but the result of glycolysis in these cells, exercise physiologists prefer not to use the term fermentation. So there the fermentation is actually a process which takes place in the absence of oxygen. But here, as discussed earlier, that the person is undergoing intense hard work, intense exercise, therefore is in need of urgent supply of ATPs. Therefore, this glycolysis process occurs very quickly as compared to the completion of the full 3 stages. So therefore, even in the presence of oxygen, the glycolytic pathway is preferred to generate ATPs urgently, and in this way, even in the presence of oxygen, lactate is produced. So therefore, lactic acid fermentation is not referred to as fermentation. During strenuous exercise, when carbohydrate catabolism outspaces the supply of oxygen from the blood to the muscles, lactate can't be oxidized to pyruvate. Means during strenuous exercise, during intense hard work, when carbohydrate catabolism outspaces the supply of oxygen from the blood to the muscles, lactate cannot be oxidized to pyruvate. So the lactate that accumulates was once thought to cause muscle fatigue during intense exercise and pain a day or so later. Usually after strenuous exercise, after intense hard work, you feel some pain, some restlessness in the body a day or two after. So initially it was thought that the culprit behind is the lactate, but recent research has shown that it is not the lactate which is responsible for this condition. It is actually the wear and tear of the muscles. Furthermore, within an hour, excess lactate is shuttled to other tissues for oxidation or to the liver and kidneys for production of glucose or storage molecule of glycogen. So it means once your body accumulates lactate, even within an hour this lactate can return back to glucose or glycogen in the liver through Cori cycle. So it means it is not the reason responsible for the body pain. Next day, muscle soreness is more likely caused by trauma to cells in small muscle fibers, which leads to inflammation and pain. So this is because of the wear and tear of the muscle fibers, which results in pain a day or so later, and not the accumulation of lactate. Now we will compare fermentation with anaerobic and aerobic respiration. So fermentation, anaerobic respiration and aerobic respiration are 3 alternative cellular pathways for producing ATP by harvesting the chemical energy of food. Once again, I am repeating these three processes. Aerobic respiration is of course the one where oxygen is present. Glucose molecule is broken down into two molecules of pyruvic acid. Pyruvic acid is converted into acetyl coenzyme A, which enters into Krebs cycle, and all the NADH and FADH2 generated in this process are used to deliver electrons to the electron transport chain, where oxygen acts as the final acceptor of electrons. In anaerobic respiration, which takes place in the absence of oxygen, there is another acceptor of electrons apart from oxygen, so the process of electron transport chain also occurs there. In fermentation, there is no acceptor of electrons, neither oxygen, neither sulfate, so the things doesn't move ahead after glycolysis, and the NADH is converted back into NAD by delivering its electrons either to pyruvate or to one of its intermediates. All these three use glycolysis to oxidize glucose and other organic fuels to pyruvate. So remember, in any organism, irrespective of what process is going on, glycolysis will occur there because it is a cycle, it is a process which doesn't require oxygen, which doesn't require a specific structure. It can take place in cytoplasm, so it occurs in almost all types of cells, irrespective of what happens when pyruvate is produced. So glycolysis only produces two ATP molecules, net ATP molecules, through substrate level phosphorylation, and in all three pathways, NAD is the oxidizing agent that accepts electrons from food during glycolysis. Food means the intermediate stages, the substrates of the glycolytic pathway. A key difference is the contrasting mechanism for oxidizing NADH back to NAD, which is required to sustain glycolysis. So all these three different mechanisms differ in the way of oxidizing NADH back to NAD. This is the difference. In cellular respiration, by contrast, electrons carried by NADH are transferred to an electron transport chain, which regenerates the NAD required for glycolysis. So when oxygen is present, of course, electron transport chain will take place, and NADH and FADH2 will deliver their electrons to the electron transport chain. Now in fermentation, the final electron acceptor is an organic molecule such as pyruvate, lactic acid formation, or acetaldehyde, alcoholic fermentation, because even if electron transport chain is not going on, if there is no final acceptor of electrons, then still the cell must bring NADH back into NAD. So then the NADH will deliver its electrons to pyruvate or to other forms of pyruvate like acetaldehyde. Another major difference is the amount of ATP produced. Of course, in aerobic cellular respiration, maximum number of ATPs will be produced because all these stages will take place efficiently. Oxygen will be there to act as the final acceptor of electrons, and then in anaerobic cellular respiration there will be no oxygen, but there will be some other molecule which will act as the final acceptor of electrons. So it will also produce a specific amount of ATPs, although which will be much lesser than aerobic cellular respiration. Fermentation yields 2 molecules of ATP, only 2, because in fermentation only glycolysis takes place and then NADH delivers its electrons to the end product of glycolysis. So the net product is 2 ATPs. In the absence of an electron transport chain, the energy stored in pyruvate is unavailable. Of course, pyruvate is a 3-carbon molecule and 2 pyruvate molecules are produced. So these 2 pyruvate molecules can still produce a significant amount of energy, but because of the absence of the process or the mechanism, this pyruvate is wasted and it doesn't produce the amount of energy it is capable of. In cellular respiration, however, pyruvate is completely oxidized in the mitochondria where a Krebs cycle takes place. Most of the chemical energy process is shuttered by NADH and FADH2 in the form of electrons to the electron transport chain. There the electrons move stepwise down a series of redox reactions to a final electron acceptor. In aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, the final electron acceptor is another molecule with a high affinity for electrons, although less so than oxygen. So when an organism is respiring anaerobically, then there will be some final acceptor of electrons, but it will not be oxygen, it will be some other molecule, and of course its electronegativity will be much lesser than oxygen. So what will be the consequences? The amount of ATPs derived from electron transport chain will be much less. So stepwise electron transport drives oxidative phosphorylation, yielding ATP. The cellular respiration harvests much more energy from each sugar molecule than fermentation can. So the 3 processes which we discussed in the previous slides, the fermentation, anaerobic respiration and aerobic respiration. So the most efficient ATP producing pathway is aerobic respiration. In fact, aerobic respiration yields to 32 molecules of ATP per glucose molecule, up to 16 times as much as does fermentation. In fermentation, only glycolysis takes place, then the end product of glycolysis is oxidized by NADH, so the net production is 2 ATP molecules, and of course in aerobic cellular respiration 32 molecules are produced, which is 16 times higher than the product of fermentation. Some organisms called obligate anaerobes carry out only fermentation or anaerobic respiration. In fact, these organisms cannot survive in the presence of oxygen because oxygen is poisonous for them, some forms of which can actually be toxic if protective systems are not present in the cell. A few cell types, such as cells of the vertebrate brain, can carry out only aerobic oxidation of pyruvate and need oxygen to survive. Other organisms, including yeast and many bacteria, can make enough ATP to survive using either fermentation or respiration. Such species are called facultative anaerobes. In yeast cells, for example, pyruvate is a fork in the metabolic road. It's a fork means when pyruvate is produced, then there are two routes to enter. Under aerobic conditions, pyruvate can be converted to acetyl coenzyme A. Then oxidation continues in the citric acid cycle via aerobic respiration. So this is the one branch of the fork depending upon the presence of oxygen. The other branch of the fork is when oxygen is not available, then lactic acid fermentation occurs. Pyruvate is diverted from the citric acid cycle. Of course, when there is no oxygen, then citric acid cycle and electron transport chain will not take place. So then pyruvate will be diverted to another route, that is lactic acid fermentation. The purpose will be to get rid of the 2 electrons occupied by NADH. To make the same amount of ATP, a facultative anaerobe has to consume sugar at a much faster rate when fermenting than when respiring. So a facultative anaerobe which can carry out this process of which cannot carry out the process of aerobic respiration. So if it desires to produce the same amount of energy as produced in aerobic respiration, then of course it will have to increase the efficiency of fermentation by several degrees. So here is the pyruvate, which can be diverted into 2 different routes depending upon the presence of oxygen. If oxygen is present, then of course it will enter into mitochondria, acetyl coenzyme A will be produced, and then it will enter into Krebs cycle. When no oxygen is present, then the alternative is to get rid of NADH either through alcohol fermentation or lactic acid fermentation. This was the end of the lecture. Thank you.